Hello, everyone. Welcome to ATC Office Hours. We're back after a while and we have a special guest today. It is Dr. Ming-Yi Chou, a assistant extension specialist and professor in turfgrass pathology at Rutgers University. Welcome to the show, Ming-Yi Chou.
Oh, hello, everybody. Thanks for having me, Micah. I appreciate it.
It is exciting to have you here. We were just in Karuizawa, Japan, and we recorded that video that many people will have seen about your interesting article about disease suppressive soils. And I thought that was so interesting. I wanted to have a longer discussion about it. And I thought ATC Office Hours is the perfect place to do that because not only can you and I have a discussion about it, but if people in the audience have a question, they can leave that in the chat and we will try to answer as many questions as we can in this show. So this will be a video, it's a live stream, and we will also make it available as a podcast. In the podcast, I will put a direct link in the show notes to the paper. But here today, if you're watching this live, I've got a couple of screens set up where I have a QR code in the top left corner. So if you haven't read this paper yet, the title is Fungicide Use Intensity Influences the Soil Microbiome and Links to Fungal Disease Suppressiveness in Amenity Turfgrass. And there's a QR code in the top left of the screen that you can scan and you can get direct access to this right now to read it. But maybe you don't want to read it at the same time you're listening to us talk. So, Ming-Yi,
That's a long title, I have to say.
it's a long title. I read that paper again this morning, and I've now read it twice. I read it in February. I read it again this morning. It took me about an hour to read. I made a few notes. I think it is a well-written paper, but for me, it's just a really, really interesting topic. Can you give us an overview, Ming-Yi, about like what is this about?
Yeah, of course. So to start with this project, actually, it so disease suppression soil is nothing new. The idea is nothing new. It's been studied in many other economically important crops, right? Um, so I always wanted to do this research. I was kind of looking for a good system, and I joined turfgrass pathology lab at Wisconsin, and then that's when I started want to put this idea into practice. And turfgrass is actually a perfect system. And in turfgrass, I was surprised to learn that there's not much of there's some, but not a lot of microbiome study at the time when I did this research work. So I started exploring this idea with Dr. Polk, and we just want to see if there is an actual disease suppressive soil. And of course, when we talk about disease in turfgrass, especially for cool season turfgrass, the number one disease come to our mind is dollar spot. Um, and there was anecdotal observations of lower disease pressure site and higher disease pressure site. And sometimes they're linked with different fungicide use or pesticide use intensities or different philosophies of management. So we thought that it would be a very good topic to explore and to really find out if there is such a thing in turfgrass system, right? So essentially, we surveyed or collected soil samples from 8 different golf courses from the Midwest and also the Northeast, where we collect soil from fairways of different histories of fungicide use intensity. Thought that they will have very different microbiome because of the management practice. Of course, there will be um, factors like soils and climates and such, but we'll only want to zoom into the microbiome perspective. That's why we did basically like a transplant method, taking the microbiome out of those soils into a fixed condition, so a fixed soil physical, chemical factors in potting mix that we made. And then we established turf grass, creeping band grass, paying grass on top of the pots that was transplanted with the soil microbiome from different sites with different fungicide use intensity,
May I
and then
interrupt?
tested.
May I
Oh yes,
interrupt?
please.
May I interrupt for a moment and let me now narrate or talk to you about what I read this morning as I refresh myself on what the article was, so we can help people to
Okay.
understand even a little bit more about this sampling. So it was 4 golf courses in the Midwest, which was like Illinois and Wisconsin, right? And then it
Yeah,
was
that's correct.
4 golf courses in the Northeast, which was New Jersey, New York, and Massachusetts, something like that.
Yes, that's correct. Yeah,
Okay.
there are
Eh,
3 states from the Northeast.
So there were 4 courses Northeast, 4 courses in the Midwest, and then of those 4 courses, 2 of them were low fungicide use on fairways, and 2 of them were like a regular spray schedule of fungicides for dollar spot control on fairways, right? So you divide them
Well,
into
that's
2.
self-claim, right?
Okay.
We divide them into 2, but that's based on their self-report, right? So they will consider themselves as low use or high use. That's just basically a self-report. But we have the record, so we actually look into them, and then I'd say people are pretty honest.
Yeah. So in this case, with that, with those 8 golf courses that were sampled, you have the records and it matches pretty well with what you classified them as. Now, I was surprised when you took these soil samples. I read they were very quickly frozen to a temperature of minus 80 degrees Celsius. And I thought, why would you freeze it that cold? I would have thought that kills things. Like you're trying to study the bacteria and transplant them. You're trying to study the microbiome and transplant it. I was surprised at that temperature, but of course this is not my specialty. Can you tell us why that temperature was chosen?
Well, yeah, that is kind of standard. You're right about it. Like minus 80 will kill some of the microbes in our sample, but the other thing is we don't really know when we'll get all the sample because some of the sites actually the supers helped us collect the samples, right? And then we all know that they're busy, and then they collect the sample. They will send it to us. We don't know when we'll collect. We'll be able to get all of the samples and then start the experiment altogether, right? So we need to keep the soil in a place where we can potentially store for a long period of time. And luckily, we put them in minus 80, right? Because soon after we get all the sample, the COVID hit, and then so basically delayed us for another 4 to 5 months before we can actually start the experiment. And minus 80 actually helps, especially when they're in their microbes native environment where there is a good enough protection around them, where they're not a pure isolator or stored in water where they're more fragile or more susceptible to the freeze kill. Right, so we keep them in intact soil, and that helps. Of course, there'll be some that will die, but I feel like it was not the best way, but it's not a bad way to preserve them.
Okay. Yeah, because you have to preserve it somehow. And then I was
Yep.
surprised also. Well, I was pleased to read just how carefully you sterilized everything. So you took sand and you mixed it with a type of soil amendment, I think turfus, something that's like used for sports turf, which might be a
Right.
calcined clay or something like that.
Yeah,
So that was
that's
all
correct.
that was all sterilized, and then the pots were sterilized, and then you even sterilized the Pencross seeds. So the Pencross seeds were treated with chloroform, and they were also treated with ultraviolet radiation. And I was like, wow, that you're really making sure everything's really clean before you transplant this soil to it.
Yeah, and also we sterilize the growth chamber. So we use UVC light to sterilize the growth chamber. The seed, I have to say, no matter how hard you try, you're not going to get rid of everything, especially for those endophytes that live inside of the tissue. We try all we can. That is meant to clear off any microbes that are already colonizing the niche, so the seed. So um, we did UV sterilization. We did chlorine gas. We also use bleach and ethanol for surface. So we use all we could try to get rid of that what we call priority effects, because we know if there is a community there, it will be hard for other communities to get in and to establish. That's the point of sterilizing everything.
And then you took 5 milliliters of each soil and you put it into five growing containers,
It's
right?
500. Yeah, yeah,
So 500, 500
yeah. By
what?
volume, it's 500 milliliter.
Okay, maybe I misread. Or the
Oh, maybe
pod
I
is
forgot because
the pod is 500 milliliters. I read five
I think
milliliters.
so. Like,
Yeah, I was just like,
oh
wow,
no,
that's a
no, no. No, you're right. We put 5 milliliters of field soils or fairway soil into 500 milliliter pot. So that's basically 1% by
yeah,
volume.
right. So you're taking a tiny amount of soil. So for each soil was basically got replicated 5 times. So there were five sterilized pots, five sterilized pots filled with sterilized soil, and then you put that particular soil, let's say from the low input fairway from the Midwest, got put into five of those pots, but it was only just by 1%, five milliliters, and then from that you're transplanting the microbiome, or that was the idea. That was what
Yes.
you're trying to do. Did you think this
Yeah,
was
that's
going to
correct.
work? Did you think it was going to work when you put such a tiny amount of soil?
We know. I mean, from 1 grain of soil, we know that we can find, in a healthy soil, we can find tens of thousands or hundreds of thousands of different species of microbes. So, you know, that amount, in other studies, has proven working. Basically capture it's a good snapshot of the environment. You're not gonna capture everything, right? But it's good enough to capture um, a lot of the diversity.
and then it and then it grows in the new in the pot with with it what would you say, it colonizes the soil in that pot, and it colonized. I learned a new word, phyllosphere, which I think is the area around the leaves.
Yep. So basically, everything above ground, that's in turf grass, that's what we call phyllosphere.
Good. That's easier to pronounce than
Yep.
verdure or verdure, which I've which I think depending on where you're from, you pronounce it differently. But phyllosphere seems to be easy to pronounce. Maybe I'll start using that word.
Yep.
All right. So
Yep.
then you grew the grass in the sterilized growth chamber, and yeah. Now you tell the story, and then I may have some more questions. But
Yeah.
go
So
ahead. Go
actually,
ahead
it
and
was not so easy to grow the grass. So we all know growing grass is tough, but growing grass on pots in growth chamber is even more difficult. Um, the procedure. So I did a preliminary experiment before actually getting to this actual experiment. So we tried multiple methods to grow the pots. Um, First to see if the sterilized seeds work, and also to see if we can avoid any other diseases. Because we know young seeding, young seedlings are susceptible to everything. You can't have too much water or too little water. You can't have high humidity because you're gonna just have a pythium. Any sort of microbes can be a problem, like fusarium, even trichoderma, and any opportunistic microbes can cause damage on really young seedlings. So we end up using the method. Basically, it's like a sod transfer method where we grew the sterilized seed on petri dishes with just water agar, right? So once those seeds germinated and over time they're somewhat mature, we then transfer the sod onto the pots. Actually, I have some picture that I can show.
Oh yeah, please. I would love to see
Yep.
that. So, if we change to that screen, you can show us the
Right.
procedure.
Yep. Yep. So basically, this is what. Can you see it?
Oh, yeah, we can.
Yep.
We're looking at
So you can see
pots.
that's basically like a sod. Yeah. So we grew the Caribbean bandgrass on water agar first, and they all work very well. So once they establish I then transfer or uh, basically reestablish this onto the pots where I inoculate with the field soil. And after we put down, it's just like all the sod people use. We top dressed and we take very good care of them with the right humidity, and then make sure the roots are going down and then in very close contact with the soil. And so that procedure took us 2 months.
Okay, so that's after you transplanted it, or that's after you planted it on the water auger.
That's after we transplanted.
Okay, so then it's growing there in the. So you've got a lot of pots. You've got 8 golf course soils. You also did prairie soils, 1 prairie soil from each region, which is like
Yep.
a native site and completely untreated with fungicides, and 1 forest floor soil from each site. Or sorry, from
Yep.
each.
In agriculture.
Oh, and agriculture too. Yes, you had 3 extra.
Right.
So you had 14 times 5 pots, right?
Plus control. We actually have 2 sets of controls.
So you had 16 times 5
So
pots.
right.
Wow, that's a lot of work. That's
Yep.
that's a lot of work. Okay, so you've got all these
It is.
pots, and you're growing this bent grass, and each one of these pots. So you've got your controls that haven't been inoculated, and then you've got inoculated with fairway soils, inoculated with high fungicide fairway soils, inoculated with forest soil, prairie soil, agricultural soil. And then what did you do?
Yeah, so I just let the grass grow, right? Let it establish, and once they establish, so we check the root growth. We want to make sure there is very close contact of root in the soil, and the grass had enough vigor. And then we mow them. Basically, we use the scissors to trim them every other day, just to maintain them, to mimic what people would do in the field for the fairways turf. And once we confirm they're ready to go, they're all pretty uniform, healthy looking. We then inoculated with Chloridia, which is a causal agent for dollar spot. So we use Chloridia jacksonii, which is a dollar spot for cool season grasses.
And you put those on rye seeds? Is that right?
Yeah. So we prepare the inocula by adding actively growing Chloridia jacksonii into sterilized rye grains. And we let them grow for 10 days. So we make sure they're really well colonized and homogenize them very well. So we make sure we have a very good start of the disease, or good start of the pathogen.
And I read in the article that you increased the Smith Kern's dollar spot pressure. you
Yes.
increased the temperature and the humidity, and I calculated that just now. So the Smith Kern's model would have been 55% after you put
Okay.
after you put the seed down. So you had a 28 degrees Celsius day temperature, 20 degrees night, 16-hour day length, and 75% relative humidity in those growth chambers. So that was a very good. So you've added like pure laboratory pharmaceutical grade Chlamydia or dollar spot causing pathogen, and you've
Yeah.
done that in a growth chamber that has very good conditions for that fungus to grow on a grass that is a good host for that plant. So you've got the pathogen, you've got the disease triangle covered, right? Pathogen, environment,
Right.
susceptible host.
Well, that is a really good job, Micah. I didn't really calculate the Koch's the Smith-Kerm index for that, but
I, I
it's just a feeling.
Yeah, it's well, I wanted to see like what was it like because you wrote in the article that you increase the dollar spot pressure by the environment, and I said, well, what would that be? So that's an
Yeah.
easy one to calculate. Cool. So, and then it proliferated because the results, obviously this worked, and it worked better than I would have expected because of course this is not my expertise. So I would think like you add 5 milliliters of soil, it's not going to have much effect. You had a massive effect because where you did not apply, where you did not inoculate the soil, so it was just a sterile soil and it didn't have any suppressive effect at all. Those had, well, you were taking, you evaluated this by digital image analysis and you were counting percent green pixels, right? And this was like less than 10% green at the peak of disease pressure for the worst treatments. And then the best treatments, which I haven't seen these pictures yet, so I don't know how good it is, but the charts were showing like 70%, still 70% coverage or 70% green.
Yep. Right. 70% or more. So this is where you can see those drastic difference. All right. So I don't know, the third to your left, from your left column, that's basically the uninoculated control, which basically the environment will wipe out all the microbes, and I'm sure there's opportunistic from the air, because we can't filter all the air that goes into the growth chamber. So there's still some microbes in there, but it's just opportunistics from the environment. And the 70 plus would be something like the second column to your left, or the fifth column to your left too. So the difference is pretty obvious to us.
Yeah,
Which, like you said, it worked out surprisingly well.
Yeah, that's remarkable. Well, that's really cool. So then you did all kinds of stuff that I really don't understand about DNA analysis. That's what I would call it. So you identified some of the main species that were present in the different soils and in the different phylospheres, which is what is inhabiting the above ground grass plants. And so we have a big effect in what the dollar spot damage is. And then you also dug deeper to look at the different species dominating there.
Right. So actually, we start taking samples of root samples as well as the phylosphere samples before we inoculate the pathogen or dollar spot. All right. So that tells us what's inhabiting that environment, and if there was going to have some effects, we know it's because of what's inhabiting in that environment. Right, before they're being challenged by disease. Otherwise, when you inoculate the pathogen, it's gonna skew the environment. So we actually took a soil, so a root, and as well as phyllosphere samples before we inoculate, and then throughout the process, throughout the disease development process, just to see the dynamics.
And I I wondered, is this something where you look at a particular species and say, okay, this appears to be suppressive, so let's patent it and develop it as a biological control?
That's what we're trying to do, but in this study, we start looking at a broader picture, a larger picture, right? So we look at the community. So we capture all the DNA that we could capture for bacteria and fungi. And with the data we got, it was probably somewhere, for bacteria, it's probably somewhere like 20,000 different species, even though I'd say species, but we're approximating species, not really actual species, because with the DNA technology we use, it was not enough to code their actual species. For fungi, it was a little bit better, probably 30 to 50% of them. We have a good idea, at least what genera they are. They're closer to the species. And at the same time, we did cultural dependent work, meaning we culture from those soils that showed disease suppressive activity. And then we got a lot of isolates out, I'd say hundreds of different isolates of bacteria and fungi. And we do a play, dual assay is what we call. We basically challenge Chloridia with each of the isolate we get from the soil that shows disease suppressing activity.
So is that some follow-up?
And
Is that follow-up work that you're describing?
yeah, that's follow-up work. We're still working on it. We have some really good isolates from the soil, and then we're also looking to the secondary metabolites of, so basically investigating what's going on with those microbes that are showing disease suppression activity or disease inhibiting activity. And we found some of them are producing antibiotics or antifungal compounds. So that is still ongoing.
Cool. I made three. What's it called when something goes across the bottom of the screen? Ticker. I took quotes from the article and I did three of them. I'm going to play them now. And the first one is from like the section right after the abstract where it's like, what's the importance of this paper? So I'm playing this, but I'm going to read it the second time it goes, because I know some people are going to be listening to this as a podcast. So I thought the three tickers that I'm going to play here, these three quotes from the article, are quite clear to me. And this is what I took from it as somebody who used to be a golf course superintendent. It goes like this. This research provides new insights into the potential non-target effects of repeated fungicide usage on disease suppressive soils. It also indicates that intensive fungicide usage can decrease the activity of beneficial soil microbes and lead to a more disease-conducive microbial environment in turfgrass. So that's a direct quote from near the start of the article when the journals asked the authors to summarize it in simple terms. So that's a pretty powerful statement to say that intensive fungicide usage can decrease the activity of beneficial soil microbes and lead. So it basically says intensive fungicide usage can lead to a more disease-conducive microbial environment in turfgrass. And I think a lot of us have suspected that. And a lot of people want to believe that. It's just there wasn't any proof of it. And I was so intrigued to read this article and see how much detail you went into to find this correlation. Because again, you're not really doing cause and effect here, but there is this very clear correlation between fungicide use and suppression.
Yeah, I think that's a perfect term to say correlation instead of causal effects that we're seeing. And it's indirect, right? Because we took the soil and we put it in pots. It's an entirely different environment. But what we're basically, in our case at least, we show that it can make the soil environment more disease conducive. But also the other information or other points derived from this paper is basically saying not all fungicides are created equal, right? So it can make the soil environment more disease conducive, but really depends on what you use and how much you use and how frequent you use. I think that's another huge. Those are other huge factors.
Right.
Not to say that
you had chlorothalonil was one. That and there was another one that starts with F, maybe.
Yeah, flazonin. So basically, we found contact fungicides are more influential because we did a correlation analysis on how disease suppressive the microbiomes are and how much fungicide are being used on those sites. And we found a significant negative correlation, meaning the more fungicide you use in the field, in the fairway, basically makes your microbiome less disease suppressive. All right, so we saw that correlation. Sorry.
Bless you.
So we saw that correlation.
Do you happen to have that slide that shows that image, that figure, with the number of fungicide applications or the amount of AI? There's a good figure on that from the paper.
Right. Let's see.
Yeah, there we go.
Yep. So that's one of the figures. So this is the correlation, right? And then that's number of AI and times application, meaning let's say if you in the whole year you only use you only apply fungicide twice, but in each application you use two active ingredients. That's 2 times 2 equals 4. So in our study, you actually use a fairway that received up to 36 AI times application. And there was some site that are as low as 6 or even 0. Right? And then, so we did this correlation and found significantly negative correlation.
Now, on this particular...
basically...
This particular chart where we're looking at fungicide applications, this is only for the fairways, right? It's not the forest soils or the prairie or the agriculture soil.
No, that's only the fairway from the eight golf courses we took golf course sample from.
Okay. This is the kind of thing where you've got a p-value of 2.2 times 10 to the negative 16th. And I always like to look at these charts and ask grad students if they like low p-values, because this is a very, very low p-value. So what that means is something's happening there as you go across the greenness level, it's not all the same. Or as you go across the fungicide application level, it doesn't all have... They're not all having the same effect on greenness. And in this case, where you get the most greenness 20 days after inoculating with the dollar spot fungus is when fewer AIs were applied per year. So I think that's... And so then that's what you write in words. Let me put the second quote up. So then you write in words, collectively, these results suggest that higher fungicide application intensity may be negatively impacting plant beneficial bacteria and fungi in the soil, leaving the turfgrass plant more susceptible to disease development. So I thought that was pretty well explained in the article.
Yep. But again, we put the term may in there just so everybody knows again, it's a correlation that we found. It's not causal.
Yeah, this is such a big subject, and you did this on eight uh, eight soils. Let me figure out how to
Right.
get our cameras back. Now 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 I make our cameras go away. Let's see if I can figure out how to bring these back.
Yeah, like 8 golf courses soil is not a lot. I always wanted to expand this research, but this is just so labor intensive, and everything has to be really carefully designed, from the humidity control to how to sterilize the growth chamber to how you germinate your grass. It's everything. It's all about details, and that's why I feel like this. First, we were lucky. We actually we put in a lot of hard works, right? Because we test every single step before we even start the experiment. But also, we're lucky to see this experiment came through and then show us a pretty amazing result that it's beyond our expectation.
That's good. I'm still trying to figure out, for those listening, I've managed to take both myself and Ming-Yi off of the video stream, and I'm trying to figure out if I can bring our cameras back because he had such an interesting chart. Let's see. We may just be talking to ourselves, which is also fine. I'm gonna show one more quote, okay? And that is
Okay.
that's from page 16 in the printed version of the article. This present study demonstrates that reduced fungicide intensity can also induce disease suppressive soil, as it potentially allows a more diverse soil microbiome to grow and facilitate the plant microbe and microbe coevolution between plant antagonistic microbes and the pathogen. And I thought this was really interesting because it turned out that the prairie soils and I think the agricultural soils weren't as suppressive as the golf course fairway soils, even the ones that received a small amount of fungicide. Is that right?
Yeah, that is correct. Actually, prairie soil performed as among the worst. Right. So I say that environment is probably, prairie probably create a very high fitness environment for the pathogen. Uh, Maybe I should backtrack from start talking about how most of other disease suppressive soils are formed.
Yes, please.
Usually, disease suppressive soil are formed by this coevolution. So coevolution meaning the pathogen plant coevolution. So when the plants kept being challenged by the pathogen, they will find a way to fight back, basically. So a lot of time, the way they fight back is to really recruit beneficial microbes from their environment and use them as a tool to help themselves out against the pathogen. But that is slow. It's a very slow process. So that's what we call coevolution. Um, it requires such a constant a challenge for the pathogen so that the plant will actually start doing something because they react very slowly. So let's say if we disrupt that process frequently by applying fungicide, we take that challenges away. Plants basically are passively uh, living in that environment without actively trying to recruit anything that will for their benefits to fend off the pathogen. Right. So that's what we call the coevolution process. It actually requires the constant challenge from the pathogen, and that's why um, basically the plant and the beneficial microbes that will create an environment that is um, that is not so fit for the pathogen anymore after uh, a long time.
And when you say a
So
long
let's
time,
talk.
when you say a long time, is this something that can I expect it to happen in 2 months? Can I expect it to happen in 2 years or 2 decades?
Yeah. So I don't know in turf. I don't really know what. I cannot give you a time, but for potato, we know, or for wheat, actually takes decades. Sometimes like 10 years or plus. So in previous studies, it depends on the crop or plant pathosystems can range from several years to decades. Right? But we know for a fact that the more frequent you have that challenge, the faster or the more possible this synergetic uh, partnership would happen.
Yeah, it would
Right.
seem that with turf grass being a perennial, because potatoes are not a perennial, I don't
Right.
think, but turf grass is always there. So it makes me think that it has the potential to happen faster.
Yeah, I would think so too. I would think so too, because that accumulated effect is stronger over time. I would think so too.
I see. I don't know if the YouTube is working because I don't. If anybody's on YouTube and can send a message in the chat and say hi, please do, and we may be able to take some questions. I do see from LinkedIn we have a comment from Karsten Marker who says we can hear you well. Karsten might be in Denmark, and I know in Denmark. I'll tell you a story, Ming-Yi. There are a group of superintendents around the Copenhagen area who are they have a study group and they're learning from each other, and what they've done is tried to find a way to manage microbiome nivale or fusarium or pink snow mold, however we like to call it, wherever we are in the world. So they're trying to control that, which is quite a it's quite a good environment for that disease there. And they've shared data with me from 2013, 2014 era when they got started with this project. And they were having 30, 40, they would go around in what they call the Tour de Fungus in the late autumn when they've already had some very good weather for that disease. And they would have 30, 40% of the turf area on greens was infected or showing symptoms. I had the chance to visit there and attend and participate in the Tour de Fungus, going to 7 golf courses in one day, going around and checking in November what the conditions were like. And it was raining, it was cloudy, it was foggy, it was very good conditions for the disease. I went in 2022 and in 2023 on the Tour de Fungus, and so now they've gone about a decade. Some of those courses gone about a decade without fungicide treatments or with minimal fungicide treatments,
Wow.
like 1 or 2 fungicide treatments for that disease over the course of the over the year,
Yep.
and we could barely find it. And so if you go take a
Oh,
one square
wow.
meter, so you go take a 1 square meter, you might find the disease like, oh, there's 1 or 2 spots per grain, maybe, sometimes not even that.
Wow.
It's just shocking. And then they
That's
showed me that,
fascinating.
and so they're keeping the same data, taking the same measurements every year. And it's shocking how much better it's been from 2013 up to, let's say, 2023. And but of course, this is real world, so a lot of things change. They change the way that they aerify. They change the way that they fertilize.
Right.
They introduce
Right. Right.
different grass varieties interseeding. So there's a lot of things
Right.
changing, but also the disease pressure just, and they would tell me, if I'm understanding correctly, that the disease pressure just goes down and down and down.
Yep. And so in that environment, I don't know how active the grasses are during the season of microthylcium patch. I would imagine there's not a huge activity, like in the middle of the spring or summer. So maybe there is, I'm just thinking, I'm guessing, maybe there is more microbe to microbe interaction going on where whatever can attack microdocium or make microdocium uh, a positive factors to create a fitter environment for them that will grow better, right? So one good example is what we call microphagy, is whatever can actually feed directly on microdocium. Maybe that those community has increased over time. I'm just
Yeah.
guessing.
That that's
Yeah,
how I would guess too. Is like I think that there's so much diversity in the soil, in the microbiome, that as you take away I think it's like the microbiome controlling itself. Like so, it's not going to allow this one disease to dominate so much because there's a lot of things that will attack that. That's what's in my head. But
yeah,
of course, that's not by an experiment. It's just the way I think.
right. And then it will be a hard experiment to design. It will have to track over time.
Well,
Yeah.
I think I'm guessing because I can see Karsten's responding in the chat that he's there on LinkedIn, but we've got nothing coming from YouTube. So I suppose because what I've tried to do on this one is stream it on both the Asian turfgrass and paste turf channels. And perhaps in trying to stream it to two channels, it's streamed it to neither of them. And I'm not going to bother trying to open a phone or a different browser to see. But I just
Yeah.
assume that we're just streaming this to LinkedIn and maybe not to YouTube. But if anybody has questions or comments, send them to us. But I'm not seeing any comments from YouTube, so I suppose maybe we're not live streaming there. But I do have something I wanted to ask you because I heard when we did that video in Karuizawa and I shared it and so many people watched it, and then a lot of people were saying we knew this already. I I I've been doing this for 30 years. I knew this already. How? Oh, wow. Okay, Nolan's here. Good morning, Nolan. He's on YouTube. So maybe this is actually working. Maybe we just when we do this at 9 o'clock in the morning Eastern time, most of our American on Maintenance Monday too. Most of our
Sorry,
American
guys.
and Canadian audience is going to be out working on the course. Um,
Right,
so
that's true.
so
Courses are closed. No play day. Maintenance
well
sprays.
hopefully some courses are closed. When you never get any closures, it can be tough to do the necessary work. So yeah, good. So it looks like we can take questions. So if anybody has questions or comments, go ahead and send them to us in the chat. But I'll go ahead and ask you that question, Ming-Yi. What do you say to people who say we knew this already?
That that's good for you because if you knew this already, that means you're sensitive to what you're doing, and you're actually taking very good care of uh, the environment, your facility, and really looking to um, what costs you're making with all the management practices, right? So I think good for you, and that's actually good for the environment and also I'm sure you probably do a very good job um, in maintaining a high standard of turf grass. And but I have to say that this area, this whole realm of microbiome, it's so complicated, and and I think we're just at the beginning of it. We're trying to prove a lot of the concepts that we feel like we know, but again, knowing versus having data to support it is still, to me, it's different. That's why I'm here doing science. Um, but I'm glad to hear people are really interested in this area, and then they can give us feedback like that. If you have a lot of, if you have other things that you feel like you knew, but I don't necessarily know, please tell me so I can design my next big project.
Yeah, that's the kind.
Oh, Micah, I cannot hear you. I don't know if it's me or it's you. Um, hopefully, hopefully the live stream still go on. Um, so I think Mike has kind of warned me in an email that
It.
sometimes his computer crashed.
Yeah. Are
Are
you
are you
there?
are you back?
Yeah, I'm
I
back.
am.
Yeah. Good.
Okay.
Well, that'll be
Good.
interesting
Good. Good.
because it said the connection was lost. So probably I was
Okay.
gone for a while.
No, yeah, for a little bit, but you're back. Good.
Awesome. Yeah. So you when I disappeared or stopped hearing you, you were saying if you know something that I don't know yet, please tell me.
Yeah. I have to say there's so much that I don't know, right? And for those people who take very good care of their turf, it's you're there every single day observing what's happening and then making that correlation in your mind between your management practices and what you're seeing, for example, disease pressure over the years, right? That's what it's hard for me to have that same experience and then grasp what people in the field are experiencing. So
So
please give me feedback. That's what we need. That's how science works. You give me the idea, and I do the work.
very good. So this was research that you said you collected the samples before the pandemic. So this was a project that happened a long time ago. And it was published. You wrote it up and submitted it in 2024, and then it got published in 2025. So you've been
Right?
doing a lot of follow up work already. And can you
Yes.
tell us what some of the exciting follow up work is that you do in the same field or same area?
Yeah, I kind of touched a little bit. All right, so we have a lot of isolates that hundreds of isolates that we initially isolated from those suppressive soil, and we challenged those isolates with dollar spot or chloridia, and actually four different chloridia species. Now we know there are cool season ones and the worm season ones, and we kept those consistent ones, those ones that consistently suppress or inhibit the growth of Chloridia species. And we look for some of them. We look into their secondary metabolites to see what compounds are they actually producing. And surprisingly, we actually see some of them producing uh, the fungicide or fungicide analogs that we know. For example, like the strobies, or even some produce like DMI fungicides. But there's so much more than that. There is no one isolate that we studied found they only produce one kind of uh, antifungal compounds, but it usually is a long laundry list of things that either is a point immune inducer or a direct antagonistic compounds like fungicides, or there'd be like something like enzyme, that will degrade, that could potentially degrade um, like the cell walls of fungi. So there are different things that going on. We did different approaches using uh, multi, multiple omics, but also we did some enzyme studies to figure out what's going on with all the isolates that we have.
And
And we also try, because the point is try to bring them into the field. So also try to apply them in the field to see how effective they are. A lot of time, you can find amazing results in the lab, but they don't always translate to the field. So this is our first year to try some of those in the field.
just
Um,
on dollar spot.
just some dollars, but I mean that's our primary focus for now, because the disease suppressive soil we studied was on dollar spot. So we're, our focus remained on dollar spot. But you know, if some of those secondary metabolites that we identified are true, we would expect that those isolates can potentially have broad spectrum disease suppression activities.
Yeah. So maybe on anthracnose or rhizoctonia or something
Right.
potentially.
Yeah. Yeah. That's what we're hoping, but we don't know before we try.
Yeah. Very, very, very interesting. So, yeah, we don't have a lot of questions in the chat. So that's, uh, that's good because we're coming up on about an hour. So we'll be here for like another 10 minutes or so. And if we don't have any questions from the chat, maybe we will wrap it up. But I
And there,
encourage. Go ahead.
oh, sorry. There's another interesting thing I can share with you. So it's not a
Yes,
direct
please.
follow up of this project, but we feel like we know too little about our pathogen even. So um, starting from 2023, we look into what we call the hyphosphere. So now we have many terminologies. We have rhizosphere, which is the the habitats around the root. We have phyllosphere, which is the habitats around the above ground leaf. Now we have the hyphosphere, which is the habitats around fungal hyphae, or we can call mycosphere, which is the habitats around um, the fungi mycelium. So we look into dollar spot hyphae to see what if there is any microbial community around the dollar spot mycelium. And the reason why I look into that is because we thought about the reasons, or we look into the literatures about the reasons why a lot of the currently available commercial biological products don't work um, as we would expect. And we found one potential reason is a lot of those products are actually derived from soil. So a lot of the bacillus products or pseudomonas products or streptomyces products, they're actually soil derived. Meaning if we took those microbes out of soil and soil is actually their home. So if we want to put anything down and then want them to thrive, they will probably only be in the soil environment instead of on the foliar environment like our turf. Right. So we're thinking, I just thought that, okay, so what would be an environment that will best suit the disease conducive environment, and that environment happens to be the greatest fitness for this biological product. And then I thought about if our target is dollar spot, then we should look into dollar spot to see what it can actually land down dollar spot and then kill them. You know what I mean? Do you follow this logic? So we actually start looking into the dollar spot mycelia and see what we can find from dollar spot mycelia. And it has to do with what I mentioned previously, the mycophagy microbes. We actually found a bunch of microbes actually directly feed on um, dollar spot hyphae.
Are these
So that's
bacteria
like
or
perfect.
fungi?
They're different kind of things. So they're bacteria. They're also yeast that we found can potentially degrade the cell wall of Chlamydia or dollar spot, and then potentially feed on them. Some of them are actually endophytic, so they live inside of the hyphae of Chlamydia or dollar spot. So that's another direction that we're going. You know, and we know dollar spot is foliar disease, so we look into the soil, we look into the root. Now we're actually looking into more directly from where the disease happened in the fungal mycelium.
Wow, that is
So
really
now we have a
really
collection of microbes.
yeah. That's interesting. So you think eventually you'll have some new products, perhaps?
That's what we hope. You know, it, after all, we spend so much time and resources looking into these. I hope there be something that not just a device, but also a product that product always sounds more practical, right? People can take out to the field and then use it and effectively help them with their disease problem.
So, what is the practical advice that you would give in terms of fungicide scheduling? Because let's say that we want to have or we need to have control of dollar spot. So, control might mean different things to different people, but it needs to be controlled at an acceptable level for a particular facility. So, based
Right.
on this research, you want to minimize the fungicide application, and you want to minimize particular types of very effective fungicides as well. You want to minimize that. So practically, what would you do to get the best of both worlds in order to optimize control and hopefully not, hopefully allow the soil to be as suppressive as it can be?
Yeah. Yeah. So I think there are two takeaways from this study that we talked about today. One is really looking, the fungicide class matters. So we basically what we found in this study was contact fungicide would have more impact on the beneficial community or disease suppressive community than um, other classes, right? So the penetrant fungicides. So contact fungicides are great tools. I'm not saying that we shouldn't be using those, especially when you have fungicide resistance, right? But think about what's the timing of using those contact fungicide. When you need to use it, you have to use it. But if you don't need to use it, you can. It's better not to use them just for the sake of convenience or for the sake of um, a peace of mind, right? Um, so there is, I mean, you can do that, but there is a consequence now we know is you can potentially wipe out your beneficial just by using one uh, applications of contact fungicide. So that is actually a a long transition. So if you need to use it because you have potential fungicide resistant problem, then you need to use it. But then you have to at least have a goal of trying to reduce that application. So you use that very specifically, and in the long run, you're building up your beneficial microbes. Maybe in 5 years, you don't need to use it. The other thing is the, f- uh, the rate we talk about in this. Actually, didn't talk about directly the rate you're supposed to use, but the rule of thumb is newer fungicide, lower use rate seems to be better. Because if you really draw a line on those negative correlation analysis, you found there is certain cutoff or thresholds of use rate per year where you can still use synthetic fungicide, but you're not going to impact that um, greenness after the turf grass are being challenged by disease. All right, so there's certain use rate cutoff. We're still trying to, fine tune that. This is not really a recommendation, but the rule of thumb or the takeaway message here is if you can use lower rate, more specific fungicide, that seems to be better, indicated by our data. If you can move away from uh, non-targeted fungicide like contact fungicide without a reason, then you shouldn't be doing that, right? So those are the two major takeaways I think uh, derived from this study, backed by data.
And you also had the nitrogen rate history from these places, but did that had an effect on? Let's talk about that for a minute because that was interesting. It had an effect on the soil microbiome, so the community was different based on what the nitrogen rate was. However, it did not have an effect on the outcome that was measured in terms of dollar spot disease severity in this experiment.
Right. Yeah, in this experiment, there is no significant correlation between disease suppression and nitrogen use rate history. Um, yeah, that's what we found.
Now we have a question. I'm going to throw it on the screen. Nolan Harker asks, would rotation of different contact fungicides still show similar results?
Well, that's a very good question. The answer is we don't really know, but based on the data we have in the fungicide use history of the sites that we sample from, it tells us using older contact fungicides, for example, chlorothalonil, it seems like one application of a full rate to control dollar spot is enough to go above that suggested rate where to maintain your healthy or disease suppressive microbiome. So it seems like one application will start disrupting your dollar spot suppressive microbiome.
Just one application just one application of chlorothalonil messes things up for a while at least.
In our data, at least from the sites that we sample. And I'm not, again, this is not fungicide recommendation. And like I said, these contact fungicide like chlorothalonil, those are a really good tool that can help manage different things, especially for fungicide resistant. But based on our data, one application would start disrupting these microbiome disease suppressiveness. So like I said, I think it's a long transitioning. If you need to use, because you have those disease problems that probably partially built up by the untargeted fungicide application, right? So if we keep this disease suppressive microbiome idea in mind, and we're going towards a direction with reduced fungicide or more specific targeted fungicide application, then maybe we can start with thinking about whether or not I really absolutely need this contact fungicide in this application, right? That's the first thing to think about, and that's a start point, right? And maybe you still need 3 apps of contact fungicide this year because you have fungicide resistant problem, or you have other problem. But with that goal, maybe next year you reduce to 2 applications, and in 5 years you only need 1. In this process, you're creating the environment that your beneficial microbes or beneficial microbiome can start building up. Right? Maybe in the goal of year 8 or year 10, there are occasions where you don't need any of your contact fungicide, where your more targeted fungicides like the DMI, QO, ISDHI will be enough, right? But again, there's so much that we don't know. This is just like ideal or situation that based on the data that we have from this paper.
It would be interesting because I'm sure you've heard anecdotes. I've heard anecdotes and different locations around the world are popping into my mind. Different turf managers that I've talked with who have, and I've visited their sites, and they've applied a surprisingly low amount of fungicide. And it's not in year 1, but they're like, even they've expressed to me surprise at how little disease they're seeing in conditions where in the past they would have said that they would get a lot of disease. And they're not pesticide free, but they're using less pesticide than they used to, being more reactive instead of proactive, and being very thoughtful about what they're doing. I think it would be interesting to do a survey, not necessarily collecting soil samples and not collecting microbiome samples from these places, but just to try to gather those anecdotes and just see, okay, what diseases have people observed this? Because if you're a turf manager working somewhere for 10 years and you're like, wow, I don't get anthracnose nearly as bad as I used to, and I'm also using less fungicides, or I don't get microbiome nearly as much as I used to, or I don't get dollar spot nearly as much as I used to. That would be very interesting, because right now I think that it's like that information is out there, but it's in this person's head and it's in that person's head, and they're in different countries, and maybe you've never heard that story. And then maybe I can't remember the story because I'm mostly interested in fertilizer and organic matter and green speed. So I hear the disease stories, and I don't necessarily they don't stick in my mind and link together like they would if a plant pathologist was hearing them. That
But
could be
I think all the
interesting.
factors that you mentioned are, all these factors will have a very critical roles, right? Because if you have a hard goal to reach, like, low disease pressure, low disease incidence, then you have to think about all those things you can use, right? The fertilizers, the fertility, and the grass cultivars, and what different kinds of fungicides you have, and the programming of all these things, and remove do removals, or all those factors matter. And like you said, it's all different ideas. People have stress on one thing, but maybe not on all the things. How do we put those information together and then have a broader idea of how we can achieve this goal together? It's a little bit tough,
We have a
but
very
definitely
good
it's a direction that I want to go.
we have a good question here from Jeff Fenn, who asks, if, for example, you had a practice green which had no fungicides applied, and on that practice green you have good resistance, it seems to be showing good resistance to disease. Could you hollow core that green and then use those cores? Presumably, it's going to be a sandy material, and could you top dress and inoculate the other greens on the site? Good question, Jeff.
I think you can potentially do that. I think you will definitely move if your disease resistant green, uh, that disease resistant actually come from the microbiome, right? Not from the fertility, not from other factors, not from like the soil structures or anything else, right? If it's actually from the microbiome, I think by taking the microbiome from your disease resistant area and put it on your other green, you will definitely be able to move your disease suppressant microbiome around. You can potentially put them there. Whether or not they establish, uh, it's a different story. So again, you have to think about multiple factors there. Is the soil structure the same, right? If their soil structures are pretty similar, or the sand structures are pretty similar, then I'd say you have a good chance of establishing your inoculated microbiome into your new area. The other thing is, if you're gonna uh, keep doing the fungicide application you've been doing that created non-disease suppressive environment, then maybe you will have a flush of activity, but then you're going to eventually suppress those beneficial microbes or microbiome with your current management practices, right? So I think inoculating your beneficial microbiome is one thing, but maintaining and then culturing, providing them the environment that they need is another story. All right, that's why I think that's part of the reason why I think some of your biofungicide don't work is because you put down one biological fungicides before those living organisms can thrive and establish in your environment. Maybe you spray something that has bactericidal effects, you kill them off. So before you even see something, they're gone. Right? That is one of the reasons why I think some biological products don't work as effective as we wanted. Does I hope that answers your question.
I have another question or observation. We see in Northern Europe, or in, let's say, north of the 45 degrees latitude, north of 50 degrees latitude, dollar spot seems to be coming up and being more and more of a problem. So there's more and more dollar spot in areas that it wasn't commonly seen before. And the outbreaks, from what I've heard and from the pictures that I've seen, are pretty intense. And I'm like, wow, I just never would have thought of those areas as having really bad dollar spot pressure. But as the disease is coming in, it seems to be causing some big damage. And the question is, if you have a pathogen coming into an area where it hasn't really been common before, would it be something like if it's going into those prairie soils and then it can easily grow, but there's not a suppressive microbiome built up? And so we would expect by theory that the severity of dollar spot should be less than it currently is. Is that
I think you're
correct?
absolutely right. Yeah, I think so. There's no data to back that, but based on our prairie study, soil sample, disease severity pretty high, or disease pressure is pretty high in there. I think once a pathogen go into a version, then there is no biological resistance. I think the disease can flare up pretty bad, and maybe that is what's happening in what you described. But there is a constant microbe to microbe arm race, as long as you allow it, right? And there is a plant microbe arm race as well, as long as you allow it, right? So that goes back to how previous studies of disease suppressive soil, how those disease suppressive soil form. First, you need to allow the disease to happen, and almost at every cases, the disease came in so strong. There's almost no way for people to control those diseases. That's how those disease suppressive cells start forming. So they just basically abandon the field, and after several years, they start seeing naturally the disease severity became lower and lower year after year. So you need to allow the disease to happen. You need to allow the surrounding microbes to do their work. You need to allow that arm race to happen without disruption, right? So I think at a very early stage where the pathogen just come into the environment where it's conducive, it can come in very strong.
And
that's absolutely the case.
And that's difficult to deal with. But if we would say there's some light at the end of the tunnel, based on theory, it's not guaranteed, but it would seem that the suppressiveness should develop in the soils. So we have another comment.
But you have to do it in a very careful way.
Daniel Noonhagen says, thanks a lot. Micah and Ming-Yi, that's right. The pressure in Northern Europe is as severe as described, which is difficult because that's a part of the world where there's not a lot of fungicides available by law. But that's why I think it's so fascinating. I'm actually going to Denmark in 5 or 6 days from now and go see some courses that don't have fungicides applied. I think it's absolutely fascinating. And this is not my area of expertise, but this is, I was so excited when I read this article because I'm like, yeah, this is finally, there's some science that shows that this is what's happening in turf grass, that it can happen in turf grass. So I think I just have a lot of anecdotes myself. I'm not so scientific on this topic. I like to read what other scientists do. But I just have like my anecdote is if I go and look at parks, like low maintenance parks, I don't see any disease. Even like fairy ring, there's like not problematic fairy ring and not rhizoctonia. and certainly none of the diseases like that really would cause it. If there is dollar spot, you don't see the symptoms, right? So all the fungi must be there, but you don't see the symptoms. You don't see pythium. You don't see these kind of problems in parks so much, in low maintenance parks. And now maybe it's because I'm seeing mostly warm season grass in tropical areas. But even if I would go to Hokkaido, if I would go to northern Japan and go through parks in the summer with cool season grass, I'm not going to see like these huge disease outbreaks. And I just think in nature, there's more of a arms race. And you described it as an arms
balance,
like checks and balances, an arms race between the good, the pathogenic fungi and the fungi, the microbiome that would actually be antagonistic or suppressive, disease suppressive microbiome to those pathogens, and it leaves the it leaves the symptoms basically much less than they are on fungicide treated turf, where it's the symptoms seem to be more severe. That again, that's very anecdotal, but I was so excited to see, yeah, there's people working on this, and then you prove it. So for everybody, read that paper. There's going to be a link when I post this video, or you'll be able to watch this video recorded on demand, and you can listen to the podcast. I'll get this posted. I'm going to put a direct link to the paper, and it's really an interesting paper, and you can look at the charts and you read the details and you're like, wow, there's really something there. From 5 milliliters of transplanting this soil, but not every soil. And the soil that you would think, that prairie soil that hasn't had fungicide, so you think, okay, this is the pure virgin soil with all the good stuff in it that you might think if you're going to go buy something, like we're going to get a prairie soil extract and apply that to turf. That could potentially be good food to stimulate the pathogen. So it's not always the way that you think. So it's really interesting.
I think it's all about balance. Yeah, it's probably all about balance. So there might be those beneficials in those soils. It's just they're not in the amount or the abundance that required to have the suppression activity that you can see. And there's so much more data, derived from this study than what we can publish, because there's always content limit for what you can publish. And one thing that we observe in these disease suppressive soil is they actually harbor more dollar spot pathogen load, but they're not showing as much symptom. So that basically kind of indirectly aligned with what you said, where a lot of time you don't see as much disease, where there's probably pathogen there, they're just not showing up. But so
yeah,
we didn't put that data in the paper.
Yeah, and this is why some people say, why don't I study or I'm always doing stuff about golf, and I'm just like so interested in golf and less so in sports turf. Like, I recognize sports turf is very interesting, but for me, like golf, we're managing a property, we're managing land, and it's perennial, and we have the potential, if we want to try to develop disease suppressive soils. And I just see sports turf as more like we have to prepare it for the game on this day, and then we're going to resurface the field so we can produce a pitch for 2 months from now or so on. And we'll rebuild the pitch every year or something. It's less of a perennial land management thing. But with golf, the people who are managing golf courses or lawns or parks or things that don't get rebuilt all the time, don't get resodded all the time, don't get replanted by seed every year, then if you're managing something that's a perennial, you can manage not only the surface, not only the grass, not only the organic matter in the root zone, but you're also managing the microbes in the soil, which can be really exciting. And probably in all of our lifetimes, even the youngest people listening to this, we're probably not going to understand we'll understand a huge amount more than we do now, but it's such a big topic, we can't imagine that we would ever understand it really.
Yeah,
But
it's a whole universe. Yeah.
yeah, it's crazy. But when we can keep understanding it more, we can study it, and if you like learning and you like learning new things, this is a universe of potential interesting study. So that's very good, and that's, I think, about all that we need to discuss on this topic. Don't you think, Ming-Yi? We've covered it pretty well.
Yeah, yeah, I think so. I mean, thanks for all the questions and all the ideas. I think this is what I need to think about what next 5 steps I should take for my future study on the microbiome and disease in turf.
What percentage of your working life is devoted to this aspect of like this research path? Is it 5, 10, 50? Because I know you do a lot of other things.
Yeah, not enough. Not not enough. I think I'm probably only my program right now. I think this is probably only 20% or 15%. Um, I'm trained as a microbial ecologist, so I obviously want to do more in microbiome. And there are many new tools that I can play around with. It just you know these kind of research costs a lot of money, because it takes a lot of time. Um, it just with the craziness of being a new or newer young faculty, there is not always enough time to really sit down, think through, and do hands-on work and do it in the way that I think it's careful enough for a meaningful microbiome study. So I think it was really a privilege that I'd be able to do this kind of study when I am with all the other researchers that at multiple universities when I did this work. So
Yeah, that's yeah. So this particular paper has a lot of other people involved, but you were the main person doing the work, and you collaborated with Paul Koch and let's see.
yeah, there are many
Here's
people. I mean,
everybody. Here's all the authors. Patil,
Dr.
Wo,
Bruce
Lei.
Park helped collect the soil samples, and Dr. Frank Rossi helped collect some of the samples, and some of the author you see here.
Yeah. So
Apruva, yeah.
yeah. So that's
And
that's cool. Well, I hope you can continue doing this kind of interesting work, and hopefully you can do some more of it. I know the person I'm going to contact when I want to know more about microbial ecology of turfgrass. And the next time that we have a show, maybe I'm going to ask you if people want to send their soil to be tested. Is that possible? And so on. But we're not even going to get into that today, because that is the end of this ATC office hour. So thanks, Karsten, Nolan, Jeff, Daniel, for joining us in the chat. I know there's a few other people out there who watched also. And I know a lot of people will listen to this. So if you have any questions, let me or Ming-Yi know. And yeah, this is exciting stuff.
Thank you. Thanks for having me again. I think I want to say the last thing is there's no fungicide recommendation here. What I'm saying is based on what the data I have. So I hope I didn't piss anybody off.
Yeah, I hope not either. I hope we shared some useful information and didn't misstate the case too badly. All right. Thanks everyone for watching, especially those who joined us live. Thanks everybody who takes the time to watch or listen to this in the future. And thank you Dr. Ming-Yi Chou, for joining us and giving so much of your time to share the information today. I will sign off now for ATC from Trong, Thailand. I'm Michael Woods. Bye-bye.